And today we are going to talk about the importance of the herbal medicine research for the whole world. As soon as the pandemic started, people worldwide started to use randomly herbal medicine, which caused a huge disturbance in the public and social media, which led people to spread and forward misinformation about the miracles of the herbs to treat and cure the infected COVID-19 patients. WHO, World Health Organization, warned people not to use herbs as a remedy to fight the virus. Therefore, the platforms of social media like YouTube, Facebook, Twitter, and other social media deleted and prevented or prohibited publishing such info on their platforms for the health safety of the users. But after a while, they gave up controlling that, and the rate of misinformation declined in that social media. Any claims of such efficacy of herbs against viral infection should be done by researchers under supervision of professionals and experts in scientific centers that are well funded by their universities. Otherwise, encouraging people to use herbs randomly without biological background knowledge of the disease and the herbs is a very dangerous behavior that should be stopped and stood against. Conferences like this one and the herbal medicine meetings of specialists should be published and focused on in the social media instead of circling the exaggeration of folk talk about the curing plants in social media. Just type miracle plants on the YouTube search bar and see how many misleading videos you are getting there. For example, Grape Miracle video mentioned the benefits of the fruit without mentioning the main effective part of it, the seeds. Without grape seeds, there are no real benefits. I found that the Asian developing countries like China, Pakistan, India, and Malaysia have published many scientific articles about the safety of herbal medicine than the developed countries like United States. Studying herbal medicine without knowing the LD50 of those herbs is a risk for human health. When a researcher experimenting on the herbal extract as antibacterial in vitro without considering the LD50 value of that herb to set parameter level of the herbal concentration, the results of that experiment will be meaningless to human health and never serve the herbal medicine knowledge. That's why I conducted calculating LD50 of 11 herbs to be starter for other researchers to follow this important path to serve the herbal medicine wisely. 4 statistical methods were used to calculate their LD50. 30% alcohol extract of each herb were prepared. 8 concentrations, 1, 2, 3, 4, 5, 7, and 8 grams per kilogram of body weight of each extract was orally administered into 20 albino mice for each concentration. Observations and notes of mice behaviors before mortality within at least 48 hours after oral administration of each herbal extract were recorded. And these 11 herbs are Al-Hajji grey crumb, agul, Epium graveolens, celery, Capparis spinosa, copper, Glycyrrhiza glabra, licorice, Nigella sativa, black cumin, Plantago lanceolata, plantain, Populus euphragtica, poplar, and Salix acmophala, willow, Tribulus terrestris, caltrop, Trigonella furum graecum, fenugreek, and Urtica urens, nettle. I found that LD50 for black cumin, celery, poplar, willow, and agul extracts were laying between 4 to 5 grams per kilogram. LD50 of caltrop, licorice, and copper extracts were between 5 and 5.5 grams per kilogram. LD50 of plantain and nettle extracts was 6.56 to 7.67 grams per kilogram. Amazingly, no mortality was indicated with the fenugreek extract, even in higher 12 grams per kilogram concentration. Therefore, no LD50 was calculated for it because it's high protein content and very, very safe. Weight loss, weight gain, and sexual drive of mice behaviors after oral administration of herbal extracts were discussed. And here is the results and my findings in this table. As you see, in table number 1, the 4 LD50 methods values estimation of the herbal extracts, which is gram per kilogram mice weight. You can see the last column, the average of these 4 methods. And it looks like that the black cumin, which we call it soda, Nigella sativa, when it's just like 4 or very close to 4 for the LD50, that means 4 grams per kilogram is very close to be unsafe. And the next Urtica urens, nettle, the Urtica urens, you can see it's very high. It is 7.67, which is very, very safe. And here is the table number 2 about the observations and notes before the mice die within 48 hours after oral administration of each herbal extract. As you see, the first 2 concentration, 1 gram per kilogram of the body weight and the 2 gram per kilogram of body weight for all the 11 herbs are safe, and there is no mortality at all for more than 2 weeks. We can see also in celery, the sexual activation happened in 3 grams per kilogram, but 2 of the mice from the 20 die. And as you see on 4 grams per kilogram, 4 from 20 die, in spite of the others still very sexual active. In 5 grams per kilogram, sexual rampage. What I mean by that, the mice, as soon as I injected or administrated the herbal extract, that concentration, 5 grams per kilogram, they jump to try to make copulation with the female, and thus make the rampage. If there are 2 males in the same cage, then they try to kill each other to get the females. And after the 6 grams per kilogram, the mortality increased, 17 of 20, and at 7 grams per kilogram, bloody urination and death complete. In black cumin, the concentration of 3 grams per kilogram, the sexual activation happened to mice, and there was no mortality. Within 3 days or 4 days, there was no mortality. But still, in my opinion, 3 grams per kilogram, it is very high dose, in spite of it is increasing the sexual drive for the mice. I don't suggest black cumin to be used for this purpose. Alone, I mean. You can mix it with other plants, very safe, to be diluted. And here is the main uses of the 11 herbs: العرقول, celery, copper, licorice, black cumin, plantain, poplar, and willow. The same uses: fenugreek, nettle, caltrop. It is in front of you, and I promise I'll upload this video into on my channel on YouTube, and you can see it, whenever you like. Finally, I hope that was useful and helpful, and encouraging you to set up your plan for the herbal medicine research, and be careful if there is no LD50 for that plant or that extract plant, you should do it, or try to find somebody to do it for you, and then set the parameters level of that concentration in any kind of study: antifungal, antibacterial, and whatever you like to do. Wish you good luck in this conference, and thank you for listening, and enjoy the rest of the conference.